J'espère que vous voyez tous bien l'écran. Juste avant de vraiment commencer, je voulais juste dire que je suis super content de pouvoir se venir en vrai, avec des vraies personnes, malgré tout. Donc merci à vous d'être là, et puis merci à tous les gens qui sont connectés sur Zoom. Donc je vais commencer et ça va se dérouler en anglais. So first, I would like to welcome and to thank the members of my jury for kindly agreeing to evaluate my thesis. It's unfortunate I can't meet you all in person, but I'm really honored to have you all in my jury. During the past 3 years, I studied the evolution of flight in neotropical butterflies from the genus Morpho. And doing this, I investigated how flight contributes to the diversification of species in sympatry. Before talking to you about Morpho butterflies, I'm going to introduce this broader framework. So my work addresses the role of traits evolution in the diversification of species. What traits evolution has to do with the diversification of species? Well, phenotypic evolution, evolutionary changes in morphology and behavior can enable novel opportunities.
Attends, ici, c'est quoi?
Into a novel habitat or in using different processes.
C'est lui, ça.
Ultimately, populations specializing into different ecological niches may become separate and creating new species, contributing to species diversification. The evolution of some traits has played a major role in the diversification of species. A very straightforward example is the evolution of wings and the ability to fly. This has opened new ecological opportunities, largely inaccessible to many other taxa. In birds, for example, strikingly different flight behaviors and morphologies have evolved with contrasted feeding habits and habitat specialization. And the role of flight in promoting species diversification is further suggested by the other taxa, where flight also evolved independently, like in mammals. Bats are actually one of the most diverse groups of mammals, with highly diverse foraging strategies and habitat found among species. But the largest adaptive gradation associated with the evolution of flight occurred in insects. There are more than 1 million of flying species of insects, and they have evolved a huge diversity of flight behaviors and morphology in association with their large ecological diversity. So in my thesis, I tried to understand how flight behaviors evolve and diversify. I asked these two main questions: how flight behavior co-evolves with morphology, meaning how morphology and behavior influence each other during evolution, and how these changes can enable novel ecological opportunities. How this may kind of unlock novel niche specialization. To answer these questions, I mostly used a comparative approach, but not all comparisons are similarly informative. If you take phylogenetically distant species, for example, they are most often strikingly different. For example, a butterfly and a dragonfly show highly divergent flight behavior and morphology. One is a slow, peaceful flyer, and the other is a strong, predatory flyer. So they have diverged in many traits over very long evolutionary time. And this makes it very challenging to identify the evolutionary processes underlying their divergence. In contrast, closely related species have generally diverged in fewer traits. So ecological differences among them are more subtle, and this makes the selective pressures driving divergence easier to identify. And to understand diversification, it is more relevant to look at species living in the same geographic area, what we call sympatric species. Sympatric species are expected to interact competitively or reproductively. And when living in the same place, these interactions can drive phenotypic or ecological divergence. But during my thesis, I focused on a group of closely related species, the butterfly genus Morpho. Those large butterflies are mainly are emblematic of the Amazonian rainforest, and they are mainly known for their blue structural coloration. But as you can see, they are also quite diversified in wing shape, size, and color. And in this genus, many species can be found in the same locality, with a maximum of 16 species in North Peru. So I asked how this close promiscuity between species affects the evolution of flight behavior and wing shape. Besides, I also addressed the evolution of wing coloration that often plays an important role in visual communication in butterflies. And previous studies have provided some insights about how morpho butterflies may have diversified in sympatry. It was shown that wing shape had diverged in the genus, opposing species with more rounded wings to those with more elongated wing shape. And this difference is higher than what we expect if the wing shape had diverged neutrally, but especially is associated with divergent habitat specialization. The rounded wing species, they fly in the lower strata of the forest, what we call the understory, whereas the elongated wing species fly higher at the canopy level. So this clearly points at the role of flight behavior and habitat specialization in the diversification of morpho. And observations made on the field do suggest different flight behaviors between canopy and understory species. Here you see a video I recorded on the field. It's an understory species flying, and you can see an active flapping flight. Now here is a canopy species flying higher, and you can see a more peaceful flight with gliding phases. But to understand how flight behavior co-evolves with morphology in these butterflies, I embraced an integrative approach that was first introduced by Arnold in the 80s. This consists in linking morphology to performance to figure out how performance is used through natural behavior and how behavior is actually related to fitness. And understanding this role pathway is very challenging because it necessitates combining quite distinctive approaches. For example, morphometrics and biomechanics to test the effect of morphology on performance, and evolutionary and ecological studies in the wild to understand how behavior is related to fitness. So I applied this framework to wing shape and flight in morpho butterflies. In these butterflies, we have these two different morphologies, rounded and elongated wings. These two morphologies can be associated with a range of flight behavior because behavior is more flexible than morphology. And as you can see, the two behavioral ranges are partly overlapping, meaning that different morphologies can enable performing similar flight behavior, while being each more efficient in performing some specific flight behavior. So based on what we observed in morpho butterflies and on theoretical aerodynamics, the rounded wings may facilitate the execution of more maneuverable flapping flights, whereas elongated wings may confer better gliding performance. We can then suppose that in the dense vegetation of the understory, natural selection will favor more maneuverable flight, ultimately selecting for more rounded wings. In contrast, the open canopy may promote extensive gliding flights and ultimately select for more elongated wings. But to test this hypothesis, I divided my work in the following research questions. First, I asked to what extent wing shape constrains flight behavior. And I did that by testing how behavior responds to wing shape alteration in an experimental study. Second, I performed a comparative study of canopy and understory species to understand how microhabitat specialization may influence the evolution of flight and morphology. In the last part, I addressed the coexistence of species living in the same microhabitat. I looked at interspecific interactions between very closely related and phenotypically similar species, and I asked how their coexistence can drive trait evolution. To answer my first question, to what extent wing shape constrains flight behavior, I used an original approach. that took advantage of what happened naturally in the wild, the changes in wing shape that are due to wing damage. Here you see on the left a freshly emerged butterfly with intact wings. This butterfly was marked and captured again 2 months later with, as you can see, substantial wing damage. Wing damage is actually very common in adult butterflies. You can see many damaged individuals when in the forest. I went in the field in northeast Peru, where I've collected Morpho butterflies, both intact and damaged specimens. And for this study, I focused on 2 species of Morpho, Eleanor and Atlas. These 2 species are nearly identical phenotypically. They both look like that. And the captured specimens, they were damaged in various proportions. So I had quite a great range of different types of wing shape alterations, varying both in the location and in the extent of missing wing area. And when compiling variation in damage across all my individuals, we can see that the most frequent damages were located at the tip of the wings and along the wing margin. And very few individuals had like half of their wings missing. But then, to quantify the flight of all these individuals, we built this large experimental cage in order to film their flight in standardized conditions. So I filmed each specimen using several cameras. Here you see an example of the kind of sequence I recorded. Then I tracked the trajectory of each butterfly from different angle of views, and by combining the angle of views, I reconstructed in 3 dimensions the flight trajectory. And to further characterize flight behavior, I distinguished the upstroke and the downstroke along the trajectory. And based on the stroke positions, I defined flapping and gliding phases. Finally, I also computed other flight parameters based on the 3D coordinates. So using these flight parameters and the shape data obtained from a morphometric analysis that I will not detail here, I tested how flight behavior responds to wing shape alteration. And I found an interesting correlation between wing shape and flight. So here, each point is a different butterfly, and the shade of gray indicates the number of wings that are damaged. And the y-axis is the flight variation, where there is an opposition between higher flight speed and gliding proportion throughout the top, contrasting with a longer flapping duration throughout the bottom. And if we plot some of the specimens, we can first see that the slightly damaged individuals show quite a similar flight as the intact one. So Morpho seem to be robust to slight wing shape alterations. Individuals in bad shape, however, they show a low flight speed, they do not glide, and spend most of their time flapping. And when looking in more detail, you can see that most of the damages here were located on the forewings, suggesting that forewings may be particularly important for the execution of a fast flight and for gliding. Interestingly, I found another pattern of covariation between wing shape and flight, but this time most of the variation in damage were located on the hindwings, and this was associated with variation in flight heights. So the more the hindwings were damaged, the lower the butterfly were flying. So this suggests that the hindwings may be helpful to increase flight heights. But together, these results, they point at different functional roles of fore and hindwings during flight. And from an evolutionary perspective, we can then suppose that fore and hindwings evolve under contrasted selective pressures. For example, attacks from predators coming either from the ground or from the air may affect differently the evolution of wing shape and flight. But then I wanted to know whether within the same wing, there is a relative importance of different areas for flight. I specifically tested whether the loss of wing surface along the leading edge or along the wing margin had similar consequence on flight. I was able to do this because I realized from my shape analysis that these different types of damages were reflected by the 2 main axes of variation, either coming from the upper side on PC1 or from the lateral side on PC2. And I found an interesting result when looking at flight speed. Flight speed decreases with damaged leading edge. but not with damaged wing margin. But this suggests that within the same wing, some areas are more crucial for flight performance, while others are relatively less important. To sum up on this first experiment, I come back to my diagram describing how morphology is related to behavior. I first showed you that large changes in wing shape, like those occurring on the forewings, significantly decrease behavioral capacity, notably the ability to fly fast and to glide. And that other large changes, like those on the hindwings, decrease other aspects of flight behavior, like the ability to climb. However, my results also suggest that flight is resilient to slight wing shape alterations. So behavior can, to some degree, be adjusted, enabling to compensate for slight changes in wing shape. Well, at least this is what we see in the unchanged condition of my experiment. And finally, I found surprisingly limited effect of damaged wing margin. So behavior can also cope with relatively large change in wing shape. And here it is worth noticing that the wing margin is also more fragile due to sparser vein network. So it is then more likely to break. So the harmless consequence of damaged wing margin on flight may partly explain the evolution of eye spots along the wing margin in many butterfly species, including Morpho. And these eye spots, they have been shown to deflect predator attacks away from vital body parts. But to conclude, being able to maintain flight abilities, even when the wing shape is changed, is likely under strong selection. And this makes flight behavior a highly flexible trait. In spite of this high flexibility, long-term selection exerted by the environment may lead to divergent flight evolution in species specialized in different microhabitats. I investigated this question in the second part. For this, I had to enlarge my sample with more Morpho species. We captured all the understory species living in the locality where I did my fieldwork. And understory species are relatively easy to capture when in riverbed, because they are also really attracted with the lure. The greater challenge, however, was to capture the canopy species. This often necessitates to climb on big rocks and to use an elongated butterfly net. We captured 3 canopy species, but out of the 4 canopy species living in the area. And when in the forest, I also brought a camera with me in order to film, to characterize natural flight behavior. From these videos, I used the temporal position of the wing strokes to compute some basic flight parameters. And all the butterflies we captured were filmed in the insectary, where I was able to quantify flight in much more detail using 3-dimensional kinematics. So here I had access to a larger range of flight parameters. The first thing we did was to test whether insectary flight reliably reflected natural flight behavior. But this was the work of Samuel Schader during his master internship that I supervised. Here we looked at 2 flight parameters, the wing beat frequency and the glide duration. And this comparison was done on a subsample of species, the one we were able to film in both wild and insectary conditions. For the wing beat frequency, we found no difference between conditions. But the glide duration, however, tended to be lower in the insectary for all species, but this was particularly significant for canopy species. So captivity conditions seem to particularly affect the flight of canopy species. And here I'd like to show you an additional data point. It's an additional canopy species I couldn't capture in the wild, so I have no comparison to show you, but just to emphasize the strikingly long glides observed in the 2 canopy species here with triangular wings. Then, using the more detailed kinematic quantification of flight, I had a closer look at this gliding flight. I looked at the glide angle, which is a very meaningful parameter reflecting gliding efficiency. So I found that even if individuals from canopy species that did not glide much inside the insectary, when they did glide, it was with a shallower glide angle, enabling them to travel longer distance for a given height loss. So microhabitat selection may act on this particular aspect of flight. But this was not the only difference I found when analyzing the kinematic data. Here is the result of PCA. So this cloud of points summarizes variation in all the flight parameters I measured in the insectary. Each point is a different trail, and canopy are in blue, the story in green. And you can see that the two cloud of points are relatively well separated, suggesting a notable difference in flight behavior. What is this difference? Well, what mostly distinguished the two groups was a more powerful flight in most understory species, contrasting with the slower and more shaky trajectories in the canopy species. And the x-axis carried the variation in the use of gliding flight, which was comparable between the two groups, at least in the insectary. But then I wanted to know whether this flight variation could be explained. Sorry, first, I tested the effect of microhabitat on this overall flight variation, and I found a significant effect of microhabitat. But this suggests that the divergence in flight behavior between canopy and understory species does not only stem from phylogenetic distance. At this point, at a divergent flight evolution associated with the microhabitat specialization. And I was totally new that then I wanted to test whether this flight variation could be explained by variation in wing shape between species. So I used both my flight datasets and data obtained from morphometric description of the wing shape, and I performed an analysis that maximized the covariation between the two datasets. And I found this clear covariation between wing shape and flight. So here, each point is a different species, and the black lines are the phylogenetic relationships among them. And what is interesting to see is that independently of these phylogenetic relationships, more rounded wings associate with a more powerful flight, whereas more triangular wings associate with a slower, more sinuous flight. But this suggests that there is a causal relationship between evolutionary changes in wing shape and evolutionary changes in flight. As a step further, we wanted to understand how the wing shape divergence between species translates into different flight performance. And we focus on this gliding flight performance. But before explaining you how we did that, just a few basics about aerodynamics. When a butterfly is gliding at a given glide angle, it generates an upward directed force supporting its own weight. And this force results from two different components. The lift acting perpendicularly to the direction of the butterfly, and the drag acting in the opposite direction of the movement. And the higher is the lift relative to the drag, the shallower the glide angle becomes. The ratio between lift and drag is a measure of aerodynamic efficiency. And this lift-drag ratio is depending on the speed of the butterfly. It also depends importantly on the position of the wings relative to the incoming airflow, what we call the angle of attack. And what interests us the most here, the wing shape. So to disentangle the effect of these different components on the lift-drag ratio and test how wing shape affects aerodynamic efficiency, I worked with aerodynamic engineers who performed computational fluid dynamics simulations. So very briefly, what computational fluid dynamics does is simulating the airflow around the wings. And by solving physical equations of fluid flow, this allows computing the lift-drag ratio associated with a given wing shape in a given flight condition. This was the work of Dario Amajori during his master in thèse. So first, to approximate this gliding wing shape, we used the top view camera from my insectary, and we constructed these in silico wings to be used in aerodynamic simulations. And here we selected 3 species, 2 from canopy, the one with the triangular wings, the one with elongated curving wings, and 1 understory species with typically rounded wings. And each of these wings went through CFD simulations, and we then computed and compared the lift-to-drag ratio over a range of angle of attack. And for the 3 species, a peak was attained at an angle of attack of 6 degrees, with the 2 canopy species showing a greater lift-to-drag ratio than the understory species. So this experiment shows that the wing shapes of canopy species confer higher aerodynamic efficiency during gliding flight. So taken together, the characterization of flight in the wild, the more detailed measurement with 3D kinematics in the insectary, and the last CFD simulations, they suggest that while a closed environment rather promotes powerful flapping flight, evolving in an open environment results in increased gliding efficiency. And something that was interesting here is that this increased gliding efficiency is achieved through quite different wing shape between canopy species. So they think that there is not one unique combination of morphology and behavior enabling to achieve high gliding efficiency. But this highlights that in response to a same selective pressure, adaptive evolution may proceed through different adjustments of morphology and behavior. Up to now, I told you a lot about gliding flight. But butterflies have quite a large repertoire of flight maneuver. And divergent flight evolution may occur in many different ways. But then we asked if microhabitat selection may also influence other aspects of flight. And one particularly important flight maneuver for flying animals is climbing in flight. Climbing in flight is highly energy demanding, but can be crucial to evade predators, or in the case of Morpho butterflies, to move across the vertical strata of the forest. So we wanted to compare climbing flight performance between Morpho species. For this, we built this setup inside the insectary. It's a dark tunnel with 3 cameras positioned at the exit, and this allowed us to constrain the flight path of butterfly. So here you see a typical climbing flight trajectory when escaping from the tunnel. From these videos, we quantified the climbing flight kinematics, including the movement of the wings. So this was the work of Nicolas Silva in his master research that I supervised. And from the kinematic data, I first computed the climb angle. I also extracted the vertical component of speed, and we measured in detail this wing width kinematics. But this was done in 2 canopy species and 5 understory species. Only the wing width kinematics was measured on the subsample, the 2 canopy species and 2 of the understory species. First, look at the flight trajectories strongly suggested steeper climbs in the canopy species in blue in the figure. And this was confirmed by the measurement of climb angle. Here you can see climb angle, it is shown per wing width, so you see the climb angle getting steeper and steeper over 3 consecutive wing widths. And for the climbing speed, we also found higher speed in the canopy species. And then I tried to understand how this higher speed and angle is achieved in canopy species. And I obtained some insights from the analysis of wing width kinematics. In particular, the climbing speed was well predicted by the speed of the beating wing. So this might be higher in canopy species, even though here we have only few measurements, so this difference is hard to ascertain. This experiment highlights something very important when it comes to draw a conclusion from such comparative study. It is often very difficult to tell apart whether what we see is about performance or whether it is just about behavior. Here, I cannot really tell if individuals from canopy species they climb better because they are more efficient in this flight mode, or because they just want to fly upward toward the canopy and they behave to do so. So to sum up on this second part of my research, I come back to the initial situation where I was before performing all this work. I was in a very common situation for research in evolutionary biology, where we see a correlation between morphology and ecology, suggesting the role of the environment in the divergence of species. So here, I think I went a bit further than this correlation in uncovering various changes in flight behavior associated with the wing shape divergence. I showed in the canopy species that there is a greater use of gliding flight in the wild, and how their wing shape confer higher gliding efficiency, and that when they are placed in an insectary, they tend to fly upward, putatively toward the canopy. In contrast, in understory species, individuals who are little affected by the closed condition of the insectary and they rather show a powerful flat glide effect. So these changes in flight behavior may have contributed or even enabled microhabitat specialization. We should keep in mind, however, that among the various aspects of flight and morphology I studied, some may have enabled microhabitat specialization, but others have likely evolved afterward, after the vertical segregation, due to contrasted selective pressure exerted by the true environment. So, partitioning through space has likely contributed to the diversification of Morpho. But Morpho, well, they are also diversified within the same forest strata, like in the understory, for example. For the last part of my research, I focused on an intriguing phenomenon. Among the Morpho from the understory, some are strikingly similar, and they live together in the same ecological niche. This is, for example, the case of Morpho Eleanor and Achilles. Maybe you remember them from the first part of my presentation on wind damage. These two species, they are sister species, and they live together in sympatry over a large range of the Amazonian rainforest. And closely related species living in sympatry, they are generally expected to diverge phenotypically or ecologically because of negative interactions among them, like competition or reproductive interference. Here, this is not what we observe. Actually, it is the contrary. The two specimens you see here are those typically found in Peru. And in these two species, but in other regions of Amazonia, like in French Guiana, the wing coloration is very different. So the wing coloration is associated with the geographic area rather than with the species. And this suggests a convergence in wing coloration among species living in the same locality. To explain this convergence, we have a strong hypothesis. It may be influenced by the predator community that is different between regions of Amazonia. So here, I symbolize the predator birds from Peru on the left and birds from Guyana on the right. And as I showed you previously, these understory species have a powerful flight, but they are quite hard to catch. But they also have this blue iridescent coloration on the dorsal side of the wing, contrasting with the underside, which is darker. When they are flying, wing beats produce blue flashes, and this is particularly striking when they fly in front of dark vegetation. So here, you can see the butterfly literally disappearing and reappearing elsewhere. And this is very confusing when trying to catch them, including for me. So birds from Peru and from French Guiana may have learned the association between blue flashy coloration and the difficulty of capturing these butterflies. But on both sides of the forest, different wing coloration may have been promoted by the local predation pressure. So living in the same place and having the same wing color pattern is likely advantageous for individuals from both species because it allows sharing the cost of predator education and therefore reduce predation pressure. But such a high similarity also has drawbacks. It can lead to reproductive interference, meaning sexual interaction between species. For example, males Eleanor may court the females Achilles and vice versa, or males Eleanor may fight with males Achilles for defending territory. And these behaviors can be costly, but they are unlikely to increase fitness. So here we first asked whether the two species interfere reproductively. And for this, we focused on the populations of Eleanor and Achilles living in North Peru again, and we worked along a small river called the Rio Chicaya. Small rivers like this one is like a highway for Morpho butterflies. You can see many individuals following the river, and all these individuals are males. So this stereotyped behavior of going up and down the river is called patrolling. Males are patrolling in search for females. But what they mostly do actually is to meet other males who are also searching for females. And when two males meet, they often fly in circles around each other. And there is always one driving the other away, like, get out of my river. We can see these behaviors as contests for territory. So we wondered if this contest also occurred between individuals from different species. And if a male meets a female, they also court female from the other species. To answer these questions, I used this device. It's a false butterfly, but built with real wings of Morpho. And there is this engine making the butterfly flutter, mimicking a real flying butterfly. Important to say, we washed the wings with hexane in order to remove any potential effect of pheromones so that we could exclusively focus on the effect of wing coloration. So here you see the dummy once placed close to the river where butterfly patrol, and you can see 4 sticks around the dummy. We used these sticks to delimitate space, and we assumed that an interaction between the visitor butterfly and the dummy occurred when the visitor entered inside this space. And I also placed 2 cameras around the setup to be sure we don't miss anything of the butterfly flight behavior around the dummy. So we categorized the behavior of patrolling males as follows: passing, approaching, or interacting. First, when a butterfly was passing by the river, that you see behind, but was not interested, it was a passing. Then, if a butterfly approached the setup, seemed interested, but did not enter into the cubic space, it was an approach. And finally, if he entered the cubic space and was flying like that around the dummy, it was an interaction. And I used the 2 cameras positioned around the setup to reconstruct the 3-dimensional kinematics of the interaction between the visitor and the dummy. So this allowed me to better characterize what actually happened during these interactions. For example, I measured the distance between visitor and dummy here over time, and the flight speed of the visitor in the color gradient. So I tested the male and female dummy for the 2 species, Eleanor and Achilles. And as you see, I also tested an exotic dummy built with individuals captured in French Guiana with their narrower blue band on their dorsal wing. And each dummy was tested during 4 sunny days in a randomized order. And overall the butterfly passing in the river to more than 1,000 in the entire experiment, I computed the percentage of approach and interaction. I first show you the result with the male dummies. We found that patrolling males, they approach to their conspecific, heterospecific, and the exotic conspecific in similar proportion. There seems to be no discrimination, at least at long range. And interesting to see here, males Eleanor seem to be more curious, if we can say, because they approach in higher proportion to any dummy, but in higher proportion than males Achilles. So for the interaction, when butterflies enter into the cubic space, we found no discrimination between Peruvian conspecific and Peruvian heterospecific. But as you can see on the right, they are rarely engaged in a contest with the exotic dummy. And this is interesting because it suggests that patrolling males are more likely to come fighting with dummy bearing a locally known color pattern. Now I showed you the result with the female dummies, the female dummies. For the approach, they were also indiscriminate. The males approached both females in the 2 species. However, for the interactions, we found that males Achilles at the bottom rarely engaged in a courtship with the wrong female, but they seem to discriminate their female conspecific. In contrast, males Eleanor, the male interfere reproductively as they court also the wrong female. But then I wanted to be sure if butterflies actually make a difference between the male and the female dummy. And I could answer this question using the kinematic analysis of flight behavior. Here you see the variation in distance of the visitor butterfly when the dummy was a female. And they spent most of their time at close range to the female dummy. When the dummy was a male, however, they tended to keep their distances. As you can see on the right, how the flight trajectory was different. And I found another interesting difference when looking at flight acceleration. Patrolling males, their approach to the female dummy following a smoothly decelerated flight path, whereas they showed more erratic acceleration around the male dummy. So what we learned from the kinematic analysis is that males Morpho are able to discriminate between sexes relying solely on wing color pattern. And this is quite impressive given the high similarity between the wing of male and the wing of female. And this result also validates that courtship and contest are different behaviors, and they should be interpreted differently. So overall, the dummy experiment confirmed what we thought was happening in the wild. Males are all fighting together as soon as they meet another local signal. So the convergence in wing coloration between the two sympatric species leads to interspecific territoriality. And these interspecific interactions can also occur between sexes. As we saw in male Eleanor, they can also court the wrong female. So these interactions between sympatric species are likely costly, and therefore strongly question their coexistence. How they can maintain in the same ecological niche with such a negative interactions between them. We finally looked at another dimension of their ecological niche, the temporal dimension. Because the two species share the same microhabitat, they fly in the same space, but do they actually meet in time? To investigate for that, we performed a market capture study during 21 consecutive days. So each time we captured an individual, we identified the species and we looked what time it was. And we marked the butterflies to recognize the individuals. Here is the results. You see the number of capture according to time of the day. And this is very clear. There is a strong temporal segregation between the two species. So even if we created interspecific encounters between using the dummy butterflies, well, these interspecific encounters may be relatively limited in the wild due to the temporal segregation. But to sum up on the mechanisms enabling coexistence between these two understory species, I come back once again to my diagram linking morphology, behavior, and fitness. I started here with the hypothesis that convergence in wing coloration reduces predation pressure, and therefore increases individual fitness. But the convergence in wing coloration also led to behavioral interferences, and these interferences, they are unlikely to increase fitness. And finally, I just discovered this divergence in patrolling time, likely limiting behavioral interferences and enabling coexistence. So I think the take-home message from this last part of my research is that considering the joint evolution of multiple traits, behavioral and morphological, is also crucial to understand how species can coexist in sympatry. And to conclude, my findings nicely illustrate how species can diversify along different dimensions of their ecological niche, either through space by specializing into different strata of the forest, or through time by being active at different hours of the day. And I think another important message emerging from my thesis as a whole is that to understand the evolution of complex phenotype like flight, there is a great interest of combining different scientific fields. I could never investigate in such detail the effect of wing shape variation on flight without using aerodynamic modeling. But to understand how wing shape co-evolves with flights, knowledge about natural flight behavior is essential, and this can only be obtained with ecological study in the field. And I'd like to end with what is for me one of the most appealing perspectives of my work, those blue flashes appearing and disappearing when Morpho are flying. I would love to investigate whether and how this enables more successful escape from predators. And I think this phenomenon we see in Morpho nicely shows how flight evolves jointly with many traits and how these traits can be shaped by various selective pressures. And now is my last slide, where I'd like to warmly thank everyone who contributed to make my thesis a truly wonderful journey. My supervisors, Vincent, Violaine, Florian, and also Ramiro. Of course, all my lab mates and all the people who helped me during fieldwork and all the cool experiments we performed. And thank you for your attention. Thank you.